Hi, welcome back to LPHumananatomika.
The third component of the cell is the cytoplasm. Cytoplasm is that part of protoplasm which intervenes between the cell membrane and the nuclear membrane. It is clear, transparent in color and has a gel-like appearance. Cytoplasm is composed mainly of 80% water but also contains enzymes, salts and organelles. The three main components of cytoplasm are cytosol, a gel-like substance, the cell organelles, which are the cell's internal active membrane-bound substructures, and various cytoplasmic inclusions or paraplasms, which are insoluble substances. Organelles and inclusions are suspended in cytosol. You can think of ice cream with nuts, fruits embedded in it. Ice cream will be the cytosol. Nuts and fruits represent the organelles and inclusions. So, it can be observed that the cytoplasm is all of the material within a cell enclosed by the cell membrane except for the cell nucleus. One of the major functions of cytoplasm is to enable cells to maintain their turgidity or swelling, which enables the cells to hold their shape. Other functions of cytoplasm are the jelly-like fluid of the cytoplasm helps in embedding all parts of the cells and organelles. The cytoplasm is home to many activities of the cell as it contains molecules, enzymes that are crucial in the breakdown of the waste, hence it assists in metabolic activities. It fills up the cells, thus enabling the organelles to remain in their position. The cells without cytoplasm would deflate and substances will not permeate easily from one to the other organelle. The cytosol is the portion of the cytoplasm not contained within membrane-bound organelles. Cytosol makes up about 70% of the cell volume and is a complex mixture of cytoskeleton filaments, dissolved molecules and water. The cytosol's filaments include the protein filaments such as actin, myosin and microtubules, to name a few, as well as soluble proteins and small structures such as ribosomes, proteasomes. The inner granular and more fluid portion of the cytoplasm is referred to as endoplasm. The inclusions are small particles of insoluble substances suspended in the cytosol. A huge range of inclusions exist in different cell types like crystals of calcium oxalate, granules of energy storage materials such as starch, glycogen or polyhydroxy butyrate and lipid droplets. Cytoplasmic organelles, literally called little organs, are usually membrane-bound structures inside the cell that have specific functions. Some major organelles that are suspended in the cytosol are ribosome granules, mitochondria, endoplasmic reticulum, the Golgi apparatus, centrosome, lysosomes, microtubules and microfilaments. Golgi apparatus, the component of cytoplasm in cell, is made up of a series of compartments and is a collection of smooth surfaced, fused, closely packed flattened membrane enclosed discs known as cisternae, together with clusters of small vesicles around its surfaces. A mammalian cell typically contains 40 to 100 stacks of cisternae. Between 4 and 8 cisternae are usually present in a stack. The apparatus is present in most of the cells but is prominent in secretory cells where it intervenes between the rough endoplasmic reticulum and the cell membrane. These cisternae originate from vesicular clusters that bud off the endoplasmic reticulum. In the electron microscopic structure, it possesses two phases, an immature or cis phase directed towards the nucleus and a mature trans phase directed towards cell membrane. In addition to flattened cisternae, small vesicles of cis Golgi and trans Golgi networks form the integral part of Golgi complex. The cis phase of Golgi receives small transport vesicles with special coat proteins which bud off from endoplasmic reticulum. The transport vesicles convey synthesized proteins from rough endoplasmic reticulum and deliver their contents to the first cisternae by membrane fusion. During this process, the transport vesicles are intercepted by the communicating vesicles of cis-Golgi network, which select whether the proteins are appropriate for delivery to the Golgi stack or inappropriate proteins are shuttled back to rough endoplasmic reticulum. The proteins which are transferred into the Golgi apparatus are modified. Within the Golgi apparatus, the carbohydrate moiety is added to the protein materials with the help of transferases and the glycoprotein is formed. From the edges of first cisterna, the modified proteins are transported by vesicular budding and then fused to the edges of next cisterns until the final cistern is reached at the transphase. After a series of processing and condensation, the glycoproteins bubble out from the transphase of the Golgi apparatus as distended vesicles. Final sorting of the modified proteins and their packaging in the form of vesicles with selected sequence of amino acids take place in trans-Golgi network. So it is the trans-Golgi network which decides the destination of packaged vesicles. Some are retained within the cytoplasm as the lysosomes, while others move out of the cell as secretory vesicles and deliver their contents through the cell membrane by exocytosis. Besides the secretory cells, the Golgi apparatus in non-secretory cells liberate cell coat or the glycocalyx outside the plasma membrane. The cell coat plasma membrane complex exerts electrostatic forces which binds the identical cells to form specific tissues. So the cis-Golgi network is the first cisternal structure from which proteins are packaged into vesicles, and the trans-Golgi network is the final from where the proteins are destined to lysosomes or secretory vesicles or the cell surface. The Golgi apparatus acts as a major collection and dispatch station of protein products received from the endoplasmic reticulum. In this respect, the Golgi can be thought of as similar to a post office. The Golgi apparatus modifies or alters or processes proteins and lipids received from the endoplasmic reticulum to create glycoproteins or the phospholipids. These biochemicals leave the Golgi by exocytosis to be delivered to different intracellular or extracellular locations.
If you like the video, hit the like button. If you have not yet subscribed, subscribe to the channel and hit notification button. Join my private Facebook community where like-minded students will support, discuss, encourage and push each other towards reaching the goal. For link, visit the description area. Thank you for watching.